All right, well, good morning, everyone. Welcome to this edition of the Bioinformatics Training and Education Program Topics in Bioinformatics Seminar Series. My name is Joe Wu. I will be your host, and I am one of the instructors with the Bioinformatics Training and Education Program, BTEP for short. And today we're very happy to have David Pott and Fabian Sadell from the ISB-CGC Gateway. It's a cloud resource here to talk to us about using this tool to perform analysis of cancer data. So if there are no questions, I will hand things off to Fabian shortly. But before really handing things over to them, I would like to mention that if you have any questions, please post them in the chat or hold it until the end. We'll try to address all the questions until the end of this talk, because I believe there's quite a bit of content that they're going to be going through. And of course, this seminar is being recorded and it will be made available on the BTEP video archive within 48 hours after the class. And we're going to pop the URL for the BTEP video archive into the chat very shortly. So with that said, I am going to go ahead and hand things over to David and Fabian.
Yeah, good morning, everybody, and welcome. I just posted in the chat. Please don't hesitate to ask any questions there. You will have time at the end to ask questions, but I will do my best while Fabian is presenting. And with that, I will turn it over to Fabian.
Thanks. Hey, I'm a bioinformatician at ISB-CGC. We're part of the CRDC. And today I'm going to try to give an overview of sort of how to utilize resources that the CRDC has made available, and more specifically, how to use some of our resources that we build out and provide in BigQuery specifically, and how to sort of use the Google Cloud to perform cancer analysis. So the outline for today is I'm going to be going first through an introduction to ISB-CGC and then the Cancer Research Data Commons more broadly. And that's in part because I'm not sure how familiar everyone on this training session might be with the CRDC. And then after that, I'll be going into the ISB-CGC's specific approach to derived data. And so when I say derived data, I'll be referring to data that comes downstream of the standard bioinformatics pipelines, such as expression counts for RNA sequencing or mutation calls in a VCF file. That's sort of what we mean when we say derived data. The next part will be I'll be demonstrating a little bit of I'll be going hands-on in the Google Cloud console and sort of how to perform data exploration in BigQuery. And BigQuery is a pretty neat tool. I like to compare it to an Excel in the cloud kind of functionality. It's a little bit more functional than that, a little bit more complex than that, but it also is, if you're at all familiar with or comfortable with performing macros or function calls in Excel, it's really not that difficult to transition to doing the same in SQL. And that'll be a part of, I'll also spend some time doing a more general demonstration of how to use the Google Cloud. So I'll include a little bit of how do I boot up a VM? How do I sort of make a project? The sort of introductory steps to replicating the same functionality in the cloud that you might have on your local HPC compute cluster or BioWolf, for example. But I'll also be mostly focusing on BigQuery, and I'll be demonstrating a couple of SQL queries just to give you a preview of sort of what this tool can do. And then after that, I'll spend some time, covering the notebooks that we have made available, and I'll take two notebooks specifically and just sort of jump through in a sort of highlight fashion to give you an idea of sort of the resources that we have available. Okay, so launching right in, the Cancer Research Data Commons is a really ambitious initiative. It's been going on for a while, and the whole idea here is that we really want to make the data that is being generated in all of these, blockbuster big projects and in just cancer research moving forward into the future in general, available to everybody, and to sort of overcome some of those more traditionally existing barriers of entry to cancer research by sort of enabling everyone to utilize the cloud and sort of giving everyone access to the same sort of resources if we can. And so a big part of this is, taking data such as TCGA or CPTAC that everyone's familiar with, analyzing them through identical pipelines, and then making those available for access in the cloud, ensuring that, of course, controlled access data are still controlled and only accessible by those that have the permissions to reduce the likelihood of PII impacts, but also to then go a step further and make those standardized pipelines available to researchers, give them sort of environments to perform their analyses in that ideally they also control themselves, and that they can share those analyses and those workflows that they then generate and the data that come out of those experiments or those analyses with other researchers, and sort of this fortuitous feedback loop of continuing to share and make data accessible to everybody. And so there's a link here at the bottom. If you want more information about the CRDC and the data commons, just follow that. There's a lot of material there. So just to give a scope of the CRDC, there's more than 354 studies, more than 134,000 subjects, more than 9.4 petabytes of data, and ongoing. This is continuing to grow. More and more computers being used, more and more analyses are being done, and ideally, this feeds back into more and more data that's available, and in the long run, insights that we can make into the mechanisms of cancer and maybe into potential therapeutics for cancer. So who are we? Who's the ISB-CGC? We're a part of the CRDC. So the CRDC is largely comprised of the data commons and then cloud resources. So there's 3 cloud resources. There's us, Seven Bridges, and Terra. And we all sort of, our component in this is to try to enable the researchers, enable you guys to do your analyses, to access the data, and sort of, pave over those barriers that you might encounter and help you overcome them. So this is a screenshot of our homepage. We have a lot of different resources. I'm going to highlight them one by one real quick. So we have a BigQuery table search tool, and this is something that we built out to sort of make discoverability of the data that we offer easier. And I'll go through this a little bit later. We have a cancer data file browser. This includes an image viewer for slide images, etc. There's a couple of databases that we host. So one of them is the Middleman database of chromosomal aberrations. Then we have the TP53 database right now still. And then we have a couple of resources, tools at the bottom here. So we have a cohort builder that we're currently actively trying to improve again. We have some links to pipelines and cancer cohort APIs that we have for more programmatically minded users. We have a set of notebook. We have a big collection of notebooks that kind of illustrate how to do an analysis and sort of give you the code bites to maybe jump in and adapt to your own needs. And then we have a method for authenticating yourself with dbGaP and enabling controlled access data in the Google Cloud via dbGaP and via ERA Commons. So if you have controlled access data permissions and you want to grab data from existing buckets that are managed by the NCI, we can help with that. Yeah. So I want to highlight real quick, this is the Middleman database of chromosomal aberrations. This is a huge database of karyotyping data. This is manually curated by Dr. Middleman, who's been collaborating with us on this for a while. It's browsable. We have a search function up here. And just to, this is basically karyotyping. You look at the chromosomes and chromosomal aberrations, for example, a missing arm of one chromosome, and then we translate this into genomic coordinates as well. We have resources that enable you to translate this into genomic coordinates that allow you then sort of, pair this with more traditional genomic data, such as RNA sequencing, for example. Secondarily, we still host the TP53 database. So this is a set of all the sort of canonically or in the literature known variants of TP53. It's a really great resource for this, well-studied cancer gene. We also have BigQuery tables for this that are accessible. And then we host CNanoLab, which is a database for cancer nanotechnology particles. ongoing maintenance for that as well. So from there, I want to jump into the data commons. I don't know how familiar you all are with these, but this is sort of the major repository for data. And so it's split into three pieces. We have the genomic data commons. So this hosts the traditional RNA sequencing, BAM files, whole genome sequencing, mutation calls, copy number variation, methylation, all those types of data sets. Then there's the proteomic data commons, which is protein abundance, generally derived from mass spectrometry, and then the imaging data commons, which host a lot of slide images, magnetic resonance images, et cetera, et cetera. And so each of these data commons have their own portals. They have you can browse the data that they have there. Generally, you can build cohorts. You can access those types of data that they have, either open access or controlled access, depending on the data type. And you can really, go to these resources, spend some time on them if you're not familiar with them. There is really a vast amount of data here that's just like begging to be analyzed and begging to sort of be used. But one of the difficulties of this large amount of data that we have, or that these resources have, is that can be a little bit daunting. It can be a little bit overwhelming, and data discovery is sometimes not the easiest thing in the world, especially if you're not familiar with the intricacies of this. And so that's where hopefully our approach to enabling data science in the cloud comes in, where we try to take, what amounts to tabular data or Excel files, and we try to move it into the cloud, into BigQuery, where the barrier to entry and the sort of necessary data munging that needs to happen is minimized. And so we specifically focus on derived molecular data. We also have metadata available, but we try to make these derived data tables available. And so we provide, tooling and examples to provide for data mining and machine learning. And we try to make it as easy as possible to share results with those who choose. And we try to provide as much flexibility of scripting and computing as possible. So if you're at all familiar with R or Python, I think we have a lot of resources for you to be able to jump in here. And if you're not familiar with it, I want to emphasize it's really not that difficult to learn. If you have, if you have some time and you sort of have a project of interest, we can definitely help with learning those tools as well, and sort of enabling your own analyses. So when I say that we focus on derived data, this is sort of what I mean. This is an overview of the data types that we have. So on the y-axis here, you can kind of see the different projects that we have. And then on the x-axis, there's the different data types. And so, we have file metadata, we have gene expression, somatic mutation number, copy number, miRNA expression, protein expression, and a little bit of acetylome and glycoproteome and phosphoproteome, and those generally come from the PDC. So this is split into two categories right now. It's also possible to access a lot of the IDC data through Google BigQuery, but this is a sort of summary of the data tables that we have available in BigQuery right now. And in general, this represents the same data that GDC also has available, or PDC on their portals. So when I say that data wrangling is difficult, this is sort of what I mean. So if this is a screenshot from the GDC portal, if you jump in there and you, for example, look at RNA sequencing data, specifically gene expression quantification from TCGA, there are a total of 25,000 files for all of TCGA. And so these are all individual files. They're open access, which means anybody can anybody can download them if they want to. But if you're trying to do an analysis, or you're trying to answer a question, for example, I've identified a gene of interest in one of my projects, and I now want to just jump in and try to figure out what does the expression of this gene look like in a couple of TCGA cancers of interest, and what does this tell me about the potential mechanisms, or does this give me any follow-up sort of experiments that I can pursue? So if you want to answer that question, you have to go to this portal. You have to potentially download thousands of files. You have to then, combine them all, collate them into a single file, load them into R potentially, or into whatever data analysis tool you use, and then you can start doing your analysis. And so it's a little bit of a burden and a little bit of a lot of effort for each individual researcher. So what we try to do is we try to do that kind of work for the researcher. This is another example. There's 80 terabytes from TCGA from lung adenocarcinoma from LUAD. So there's a ton of data here, and a lot of it isn't necessarily immediately accessible to the researcher. So what we do is we have our own extract, transform, and load pipeline, ETL for short, where we take the data from GDC. We kind of do all those steps that I was just talking about. We download all of these individual files. We generally work on a program by program basis. We load them into the cloud. Then we convert and we standardize the file formats. We QC a little bit, but generally a lot of that is already done by GDC. We collate them into one large table. We create one large file for BigQuery import. We add on a couple of metadata files, such as aliquot ID or case ID, file ID, aliquot ID, sample type, those sort of fields that are necessary and useful for doing analysis right away. And then we upload these final BigQuery tables into the cloud and make them public for researchers to access, so long as the underlying data itself is also open access, obviously. And so we've done this for 25 plus data sources and programs, and for a lot of different data types, going back to the table I showed earlier. And this is a quick illustration. I mean, really, it's just we take all of the cases, the case files from a single program, and we combine them into one large BigQuery table. And so the benefit here that I want to highlight is that BigQuery is a really fancy tool. It enables simple and efficient links between these data types as well. So we have RNA-seq data, we have clinical data, we have annotation data, and we have potentially as well protein abundance data. And you can combine those all. You can join those all. Even though these tables are potentially like hundreds of thousands of rows, in BigQuery pretty simply. In R, this kind of operation generally takes a long time, especially as the data complexity goes up. Anybody that's ever tried to do a data frame join in R knows that these things can get out of hand really quickly. But BigQuery is a very unique tool that sort of scales tremendously well for these sort of applications. And then maybe even you want to use TCGA as a gold standard comparison for some data that you've generated yourself. That's also possible in this BigQuery framework. You can upload your own data into a BigQuery table that you control, and then you can join it to the TCGA data and run statistical analyses really quickly. And so just to demonstrate what that looks like, and to give you a little bit of flavor of what the SQL looks like, this is a demonstration, or this is sort of an example of me doing this in pseudocode at the bottom here, where I'm joining, in this case, the first one is an RNA sequencing table. And so this is the sort of syntax that's common for a table name in BigQuery. And then the SQL always follows the same sort of format where you select a specific set of fields. And so in this case, those fields are the case barcode and the FPKM, upper quartile unstranded. And then you select it from a specific table, and then you can add conditionals if you want. For example, if you're only interested in, if you want to eliminate zero values, this is where you'd say where FPKM upper quartile unstranded is greater than 0, for example. Or if you want a specific TCGA project only, then you could add a conditional here as well for that. And I'll go a little bit more into depth for that later. And then we join this to the clinical data to maybe get lung cancer. How many packs of, or how many cigarettes per day did the patient smoke, for example? Maybe that's a clinical field that you're particularly interested in. And so you can just join between the RNA sequencing table and the clinical table. You'd use the case barcode as a key file, key field here. And then you expand on this query. For the third one, maybe we want to do a comparison of how does RNA expression compare to protein abundance, right? And so finally, you can join this onto a protein expression table, also from TCGA. And now you've got a nice little data set, of 378 cases in this scenario that have the upper quartile stranded, the FPKM values. They have a clinical characteristic or two, and then they have protein abundance as well. And so you can see that now these are all in sort of a tabular data format together, and they're ready to sort of ingest into an analysis of your choosing, pretty much. So I just want to emphasize with this that SQL might seem intimidating, but if you are at all, if you're a scientist, I promise you, you can learn it in half an hour or so. You can become proficient with SQL in half an hour. It's not as daunting as it might seem. So, this is kind of a demonstration of a multi-omics analysis, and the CRDC in general has a pretty, and is a great resource for these sort of multi-omics analyses. So we did a little bit of analysis where we went in and we did a join between the three different commons to see sort of what overlap there is between cases. This is a little bit old at this point, this slide. I need to update this, but I think this was a year ago or 2 years ago. There were about 503 cases that had overlap between GDC, PDC, and IDC. This was mostly TCGA and CPTAC at the time, but this, as the three data commons continue to populate their resources, this is continuing to be built out, and there's more and more of these sort of multi-omics data available. And ideally, this is a great call to action for researchers to utilize these and to sort of do these multi-omics analyses that we're all, I think, really interested in. And so, why BigQuery? What is the benefits of BigQuery? How does BigQuery sort of like compare to the other analysis tools that you might have used or that you might be familiar with? So the way that Google has architected this is that it automatically scales up the number of processors for each of the tests that it runs. So it's a dynamic function. And oftentimes you can end up running statistical tests, for example, much more effectively in BigQuery than you could on an HPC cluster. So we did a little bit of a test to demonstrate this. We ran Spearman correlations and t-tests on one of our BQ tables, and we kind of just replicated and did a whole ton of these tests to see how the time scales and sort of how the cost goes up or down. And we did 2.5 million tests, Spearman correlation tests. They ran in less than 50 seconds or so. T-test, less than 20 seconds. And overall, we did another analysis where we did 6.6 billion correlations that we ran. I think those took about 20 minutes to run, if I remember right. But the total cost of running these 6.6 billion correlations was only $1.16. So the way that Google monetizes BigQuery is that it charges you for the amount of data read. And so, there's a little bit of like a loophole here where you can end up doing a lot of tests and a lot of compute for relatively cheap if you're just not reading all that much data in, if it's being performed on the same data. So it's a really neat tool that sort of has a niche that is unique. Like if you were to try to run 6.6 billion correlations on a traditional HPC, I'm pretty sure you'd be sitting there for weeks waiting for the result to come out. But you can do the same sort of analysis in 20 minutes. And so there's a lot of, I think, questions, research questions that could be answered in BQ, using BQ as a tool. And also in general, you get a terabyte of queries for free. So that means you can read a terabyte of data and query that for free every single month on the Google free tier. Generally, there's a lot of horror stories about the cloud backing up costs faster than the researchers sort of realize. And that is definitely still a little bit of a possibility, but there's a lot of ways that BigQuery is significantly cheaper than alternative methods for especially data discovery and for these sort of smaller data sets. Overall, even though an RNA-seq table might be millions of rows in the terms of how much data is that actually, it still ends up being only a couple of gigabytes, maybe a maximum like 100 gigabytes, which means you could query that table 100 times a month and not run over the free tier. And then once you're over the free tier, I think it's $5 per terabyte. So it's still overall very affordable to do this data discovery in BigQuery. So we think it's a fantastic tool. I also kind of think that it's a really cool tool for labs themselves to use to organize their data. And hopefully I can convince you of that in a bit. But the other component that we have is we've worked with some of the Google statistics. There's this cool blog by Ross Thompson and Boris Aguilar, who's part of our team as well. We collaborated on that. Check this one out if you have some time. Doing statistics and sort of bigger data analysis in BigQuery is actually not that difficult, in my opinion. We also provide, I want to just touch on this. We do support pipelines as well. I've been talking a lot about BigQuery, but you can boot up, if you're comfortable with Bash, and if you're comfortable with command line coding, and you like running your own, SAM tools or star sequencing tools, if you know how to run your pipelines, we do have a little bit of support of how to sort of boot those up quickly in the Google Cloud. We have some resources here for you to use, and we're always, we have office hours. I'll talk about those later. I'll mention those later, but we're always here to help get you set up. And we also provide a little bit of funding. If you have a project that you're interested in doing, we provide $300 of cloud credits to get you started, to get you rolling. We give you a project, we give you that budget. You can use that however you want. We give you sort of control of the project and let you sort of run with it and try to help you out wherever possible. Okay, so from here, I'm going to jump into the browser. I'm going to spend some time there before I come back to the presentation. There's always dangers of live coding here, so bear with me. But I'm going to start by going to our website. Let me maximize this. So we're back here, and then I'm going to go to our BigQuery search tool. So I talked a lot about the tables that we have, but I haven't given you a good sort of idea of what there actually is. So we have about 1,000 tables or so. We have a whole bunch of different criteria that you can filter on. For example, I've been talking a lot about TCGA, the biggest one, the blockbuster project that everyone's very familiar with. So you can select the program, then you can select data type. So for example, let me go to copy number segment. Oops. I think this is, yeah. So there's some masked copy number data for TCGA available. So we still have a little bit of HG19 data, the sort of legacy, but most of the new stuff obviously is HG38 mimicking GDC. And you can preview what this table sort of has. So this is a list of the columns that the table has, generally referred to as fields in BigQuery, as well as a description of the project. And then you can click, there's a little preview button over here as well. And you can see what the data ends up looking like. And so just to reemphasize, this is basically a tabular data table, right? This is just like an Excel sheet in a lot of ways. You can't edit it, obviously, individually per column, but in terms of browsing it or looking through it, it's very similar to that. And then we have a little button here that takes you directly to the Cloud Console. We have a little bit of a warning, but this is the Google Cloud Console. And you can see that the same sort of, or over here, there's the projects. I have the ISB-CGC BQ one already pinned. This here is our project that we store the data in. Sorry, give me 1 second. And you can see the same sort of preview that was present in our BQ search tool is also present or also available in this Cloud Console. As long as you have a Google ID, you can log into the Google Cloud and you can preview all these tables. Or you can associate, for example, your NIH ID with a Google account. You can create a Google identity for your NIH email, and then you can use that as well to access the Google Cloud. So there's some details about the table. So in this case, this is copy number segment masked again. This whole table is about 1.2 gigabytes total. It's 5 million rows. And then let me jump down here. So we have one data set per program. TCGA is right here. And we have 2 sets. We have the versioned and the sort of current is what we refer to it as. The version, this is an old, this is a sort of a backup, an archive of the previous releases that GDC has done. And then we have the sort of the singular one. So there's a lot of methylation tables, but let me jump to RNA sequencing, which is right here. So we have the field names, FPKM, TPM stranded. This is the count unstranded. So the RNA-seq table is a little bit bigger. It's about 240 gigabytes, 700 million rows. But this is sort of the strength, in my opinion, of BigQuery. This is one table that contains the expression values of every single case in TCGA for every single gene. So you can see the gene name here. You can see we have the ensemble gene ID, and then we have, this is raw counts. This is, first strand count, second strand count. And then we have the TPM, the FPKM, and the FPKM upper quartile normalized values as well. And then we have the sample type. So it's a big table. It's a little bit overwhelming, but it really enables a lot of very sort of quick analyses being done. So I'm going to jump in and I'm going to show you a couple of queries. I have them on the other screen here. I'm just going to copy and paste them. So this is, my very sort of introductory example of how to do, a cohort query or a clinical-based query. Say you're interested in looking at specific cases in TCGA that are, within a certain age range. So, this is always the format. We have select, and then we have to provide a couple of columns that we're interested in. And you can always look those columns up by jumping back here to the schema or by using the preview at the top here, project short name, case barcode, sample barcode, et cetera, et cetera. For clinical, the submitter ID is the equivalent of the case barcode. Demo, so this is demographic age at index is, the age. And then I'm also, adding vital status, for example. So this is pulling 3 columns from the table TCGA versioned clinical GDC R31. And this is one of my conditionals. So I'm saying, tell me, or return to me all the cases that are between the age of 30 and 80. So
Is it possible for you to increase the font size on here?
Yep, will do, sorry.
Thank you, sir.
So there's a total of 10,000 or so cases that fall into this range. So you can see, this executed in like 10 seconds, right? But maybe we're also very specifically interested in only lung cancer. So I know this off the top of my head. TCGA LUAD is one of the lung cancer projects. Oops. Project short name. Sorry, I have to look this up. The dangers of live coding, as always. I think it's project ID. Sorry, I should have prepared this a little bit better. There we go. Okay, so now we only have 476 cases, and I can even, just to double check this, I'm going to add that same column. I'm going to return it as an example. I'm going to rerun this query. And now, you can see that they're all project LUAD. So this is, one way to sort of quickly subset, and we have the demo with the vital status there as well. So that's a really introductory query to sort of get you started. It really is quite simple in terms of, you just jump back and forth between the sort of schema and the query that you're interested in, and you can get to a quick cohort. And the nice thing is this is dynamic, right? This is an iterative process. You can sort of see how setting different characteristics affects your total N or your cohort size, and then you can kind of iterate through. So I have a second query here. This is going into the RNA-seq data. So, the hypothesis here, or the question here is, I have a specific gene that I'm interested in. I want to know what the average FPKM of this gene is. So this query is going to calculate the average FPKM value of this gene across every single case and every single aliquot in TCGA. This isn't necessarily like a representative query that one would run, in a given project, because it's not really very informative, but it's kind of in part to demonstrate to you the speed at which BigQuery runs. So this is, you can think of this as sort of, we have a data table of, a couple of 100 million rows, and it's going to go through and it's going to select all the instances where the gene name is CCNB1, and then it's going to average all of those together. So it's, potentially like 20,000 or so values being averaged. Oops. I'm going to execute this query. It runs pretty quickly. I think it's less than 10 seconds or so. And it's going to return the total number. Oh yeah, sorry, it groups it by a sample type name. So this is the average FPKM of the gene CCNB1 for every single case in TCGA, depending on the sample type. So this group by is basically like a group the rows up by whether or not they are but by the different sample types that they are. And then, it just does the average. And, we can even remove this gene name and then execute this query across every single gene. I do need to add gene name here and then group by. So the first demonstration I did was a single gene, and now it's going to do it across all the sort of 60,000 quote unquote genes, including, putative or undetermined genes. So in part, I'm trying to demonstrate, scale here as well a little bit. This is a pretty complicated query, right? This is 415,000 averages, but it did it, and it has it by sample type name. And so now it's kind of calculated the average FPKM value of every single gene for every single sample type in all of TCGA. So it scales incredibly well, and it sort of allows a lot of analyses that would previously be sort of prohibitive.
Yeah, Fabian, we have a question in the chat. Can you point out how you browse the fields that are available to be queried? Show the method to do that.
So, like I said before, basically you select a table from the side menu here. Let me pull up this DNA methylation table here, and then it'll when you click it, it'll pop up the schema. So at the top here, there's schema, there's details, and there's preview. So schema is one way to see it. All of these fields are ones that you can query. But I prefer using preview just because it gives me a better idea of what data I can expect from the different columns. And the same information is sort of here. So schema gets you a description as well. So for example, original TCGA case barcode, and then an EG. But if you go into preview, you can see it as well. And up here, this is basically the name of each column, right? So these are the fields that you can query on each table. I hope that answers the question. So the last query that I have here, this is sort of demonstrating the same sort of join that I demonstrated before. And let me zoom out a little bit here. So this is where things get a little bit more complicated, but it's still, trust me, it's very manageable. I'm selecting fields, but now you can see that there's a little bit of like a, I'm specifying which table I'm selecting from. So the overall syntax for a join is you have select, then you have from, and this is your first table. And then you can kind of give the first table, a shorthand here. So in this case, this table here, which is masked somatic mutations, I'm shorthanding to mute. And then the RNA-seq table here, I'm shorthanding to RNA. So it's select, from, table 1, join, table 2. And then you need to specify the keys. So on, and in this case, I'm making sure that the sample barcode equals the sample barcode of the tumor, or a sample barcode tumor. So, a mutation table has two entries generally, one for tumor and one for normal. And so in this case, I'm specifically interested in the tumors. And then I'm also doing the gene ensemble gene ID equal to the gene field. So, the RNA-seq sample barcode needs to be equal to the mutation table sample barcode, and the RNA-seq table ensemble gene ID needs to be equal to the mutation gene identifier field. And then, just to shorten it a little bit, I'm only looking at TCGA BRCA. And so then I'm pulling the case barcode from the RNA table, the sample type name from the RNA table, the gene name from the RNA table, the mutation. This is like, I think, I'll pull it up in a sec, but I think this is the effect of it, and then the FPKM value. So I'm going to execute this query. And so what this is doing is it's joining between the two tables and then sort of subsetting and filtering out only things that I'm interested in. And here, so this HGVSC is sort of the, it's a shorthand for the type of mutation. So this is a C to T mutation at codon 702. And then, now I have a table which has a mutation type in a specific sample along with the expression value. So if you wanted to look at, how different mutations affect the expression of a gene, this is how you can sort of pull those data out of TCGA, out of our BQ ecosystem to do that type of analysis. And from this, you could go into R, you can generate a bar plot of some sort, or do some sort of linear modeling if that's what you're interested in. So that's hopefully a quick little introduction. I don't have a lot of time to go into detail and sort of like, we have a whole bunch of notebooks that demonstrate how to do these types of analyses. I'm going to show them to you in a bit, but I also want to spend a little bit more time jumping around the console to sort of give you a little bit of familiarity with what's going on in here, so that if you do want to try this out yourself, you're not, floundering and you're not completely lost. So the first thing to do if you're joining the Google Cloud, or if you contact us and you have a specific experiment that you're interested in doing, we can provide $300 and a project for you. But if you don't want to do that, you have to create your own project. It's pretty simple. You just have to have a Google identity. You log in, and then you can create a new project. So I'm going to say demonstration project 2, because I've done this previously. So I'm going to create, it's creating a new project, and then you have to check up here once this is done. So select the project, and this is up here is where you select the project. So, just to duplicate that. But overall, the Google Cloud Console has a lot of tools available. When you first join this, if you are joining this for the first time, it won't look quite like mine because I have these pins. It'll look more like, is this not? They've changed it on me, sorry. But there's a whole bunch of APIs here that you can see. So generally, I recommend Compute Engine if you are interested in using VMs. The cloud storage, this one is for buckets. So for example, all of the sort of TCGA files, or sorry, the GDC files are stored in Google Cloud buckets. And you can make a bucket of your own, so you can copy from GDC buckets into your own bucket. BigQuery, this is the tool, obviously that we're very sort of invested in and we use a lot. That I generally pin, and then there's another one which is Vertex AI, which is really nice. This is where you can boot up Jupyter environments, that kind of stuff. So once you're here, let me jump to Compute Engine. This is where you can boot up a VM. So as on a new project, you have to enable this. Doesn't take too long. And in general, it's very forward in terms of cost. It's very clear to you. There is a small VM, a very tiny VM that you can boot up for free on the Google free tier. While this is going, let me look that up real quick for you. So there's a section of this Google Cloud Console that can be used for free without paying it. You do need to provide some sort of some source of funding. Google Strides is an initiative that the NIH is interacting with them. You can get sort of a payment method through them, so you don't have to put down your own credit card. But like I said, there's a terabyte of queries free per month. You can have 5 gigabytes standard storage free per month. And then this E2 micro instance, this is a very small instance, but this is sort of a VM. But you can do some sort of personal project or personal, I used mine as a web scraper for a bit for sort of a hobby project of mine. Sorry, let me just jump to the other project which already has it enabled, or should already have it enabled. Okay, so this is what the Compute Engine looks like. Oh, sorry. Here. So there's a big, button up here, create instance. So if you're at all familiar with Bash scripting, or you run pipelines on BioWolf or something, this is basically the same thing. You can boot up a VM and you can run your workflows on here. You can install pipelines via GitHub or via Docker, et cetera, et cetera. And there's a lot of variability here on the types of compute you can do. So these are different costs depending on the resources that you need. There's compute optimized ones. So up here, it's giving you an estimate of the total amount per month. And just to emphasize, this is only if you're running it the entire month. So it's about 17 cents per hour. And you can turn them on or turn them off depending on your usage need. And you can more specifically, you can modify how much you need here. But there's a wide range for different purposes. You do have to be a little bit careful. You have to turn them off after you finish using them. Kind of got to remember that. But there's very cheap VMs. I mean, $25 per month is not really that much when it comes to research costs. But so this is the VM environment. This is cloud storage here. This is where you have buckets. You can create a bucket, and then you can upload files to that bucket. And then what I also want to emphasize is this Vertex AI. And they have a really cool set of sort of custom VMs in here as well, where you can boot up Jupyter environments and sort of execute code in there. Sorry, I haven't. They do change it very frequently, but this is a pretty cool tool. If you're at all interested in machine learning or you want to pursue that, there's a lot of resources from Google available for that in these Vertex AI tool sets. So the last thing I want to kind of demonstrate is in BigQuery, like say you are on your own project. So I have one here. Say you want to upload a table, right? So I'm going to create a data set here. I'm going to call it 2 because I've done this before. This multi-region here, you can, if you're working with EU data, for example, you can select a European one, for example. If you have specific needs for your data has to be stored in a specific location, that's how you do it. So I'm going to create US. I'm going to create a project here. So now this is, my very own data set, and I can create a table in here, and I will do an upload table, and I have one prepared. Go to downloads. I have an expression table here that I sort of pulled from one of our own. The file format in this case is a CSV. Table name, my data test. I'm going to do a schema auto detect, and then I do need to go to advanced options because I believe it's comma separated. I click create table. And so this is just, an example of uploading your own data to the Google Cloud. Just to pull up this. Just for the sake of time, I did already upload this on a separate project. So it's right here. This is what that ends up looking like. So, it just uploads a table. So the nice thing about this, I think for sort of organizing your own data is, we've all run into that sort of issue, I think anyway, of a researcher or a grad student in a lab does a sort of analysis. They do an experiment, they generate some data, some RNA sequencing data. They store it in a CSV table on a drive somewhere. And then, a year or two down the line, a new grad student joins the lab and they come up with a related but different question. And the PI goes, hey, we still have this old data set lying around. There's no reason we can't use that to sort of do an initial sort of analysis of if this is a worthwhile experiment to pursue. Why don't you go and get that data from that grad student and just run it really quickly? And then, inevitably, the grad student has maybe left, has moved on to something else. It's stored in some folder on a personal drive somewhere. They're all named some variation of _final _v3 _final, right? This system kind of is a sort of centralized system. So if a PI makes their own project, they have sort of top-down control of it. They can set, permissions for the students, access permissions for these tables. They can add students whole scale to the project, et cetera, et cetera. And if you're, interested in sharing one particular data table, you can just jump in and you can add, a new viewer, for example. You can give somebody permission to view a very specific table individually just by adding their Gmail address right here. And then you just give them viewer, of this table. So, if I were to do myself here, this would be basically, and then I would save it. So I could so you can assign very granular level permissions on a table by table basis, on a data set by data set basis, or on a project by project basis. And, it's all kind of automatically version controlled. there's a lot of like metadata information that automatically gets taken care of, like when it was created, when it was last modified. And so I think it's a really neat sort of interface or data storage solution for sort of bypassing a lot of those common sort of lab pain points, I think, that you encounter while doing research. Okay, so that's the general review of sort of the BigQuery. I'm going to jump back to the slides, and I have two examples of notebooks that, sorry. I'm going to jump to our web page again. So I would just want to briefly touch on this. I'm running out of time, I'm aware. We have a lot of notebook resources. So this is sort of the main sets of notebooks that we have. So one is how to get started in the Google Cloud, which goes over some of the stuff that I just went over. How do I plot a BigQuery result, for example, in R or Python? How do you join multiple tables? How do you create a complex cohort? So we have a lot of these sort of standard use cases that you might encounter like mapped out for you. So if you need a reminder, if you need like a cheat sheet, right? These are sort of good resources for that. How do you compare protein expression, gene expression in CPTAC? And then we have some more advanced things, like how do I run an ANOVA with BigQuery? How do I create randomized subsets of patients? How do I identify cancer pathways from Reactome databases? Sort of getting into the more nuanced ones. And this is one that I'm going to highlight after this, sort of how to compare gene expression in tumor against gene expression in normal tissue. And then we have statistical notebooks as well. So this is also, you can run statistical tests, specific tests directly in BigQuery. And you can even encode your own statistical tests if that's something you're interested in BigQuery. I think it's Java is the main language, and it'll execute those in a query. So, there's a t-test score, a chi-square test, Fisher exact, Spearman correlation. You can all run those natively in BigQuery and sort of utilize the sort of scaling functionality of BigQuery. And then we have a couple of introductory machine learning notebooks if that's something you're trying to pursue. And I'm going to be demonstrating one of them really quick in the slides. And so, like I said, this is a very top-down view. I'm not going to be going into detail here. Unfortunately, I don't have the time for that. But this is our sort of differential expression analysis notebook. So I based this off of an existing publication where they did the same sort of analysis. The main rundown was they took the 2,000 most variably expressed genes, and then they did a differential expression analysis looking at whether or not those genes were differentially expressed in tumor versus normal samples. And so this is, the main query. There's a couple of chunks here. So this first one goes in and selects only the protein coding genes from the RNA-seq table. So I alluded to 60,000 earlier. This is only about 20,000. And then it, for example, this is another section where it pulls all the cases of TCGA that have both solid tissue normal and primary tumor, for example. And then, sort of like it scaffolds a query to sort of whittle down the data to the final sort of data set for the analysis. And then, it loads them into R. It does a pivot into a long form, into a count matrix. And then it, runs. I have both EdgeR and DE-seq2 in this notebook to sort of do this differential expression analysis, and then plots a scatter plot of fold change versus counts per million. And the link for that is at the bottom here if you do want to take a look at this and sort of try to replicate it yourself. And then the second notebook that we have that I want to sort of highlight here, this is a machine learning notebook to predict survival from TCGA gene signatures. This is a very in-depth notebook. It has a lot of graphics along the way. I didn't write this, but it splits the training data and testing data randomly between the two. It runs the parameter optimization, normalizes the data a little bit, selects features, trains classifiers, tests them, and then has a prediction at the end. And so, like before, a lot of this ends up happening in BigQuery. So this is, the sort of two steps: selecting the data from BigQuery for the ML algorithm, and then finally, it trains the model. And this is utilizing the actual BQ ML support thing. It trains this model and then sort of, plots the classification or the clustering of those of the tumor and normal data in the training data. And then, there's in the notebook itself, there's a lot more detail, the area under the curve, et cetera, et cetera. But that's just to give you a short sort of preview of what our notebooks are like. So what are the benefits of working with ISB-CG in the cloud? There's multiple specialized databases. There's easy exploration of existing GDC and PDC data, or at least we try to make it as easy as possible. You can access virtual machines and control data for customized pipelines. BigQuery is a fantastic tool that has a lot of sort of use cases in terms of scaling Excel functionality. It lets you organize your data. It lets you share your data. It's very affordable in terms of storage. Lets you do quick statistics and sort of like get answers to those like niggling questions that your PI might have for you that you need to get an answer for quickly. And then, you know, it lets you link between data types, which is very quickly as well. It's not like a big process of sort of joining these tables together. And then there's even support for advanced statistical analyses using Python, R, and Bioconductor. And you're able to expand into machine learning if that's something that you're interested in doing. We have office hours, and this is also on our page in our documentation. These are every week, Tuesdays and Thursdays. And you can always email us at feedback@isbcgc.org. We're very willing to help to work with you to get your analysis started. And this is our team. Thank you for your time. I have like 3 minutes for questions, or if people don't mind staying a little bit longer, we can answer some questions.
All right, well, thank you, Fabian, for the excellent talk. And I guess as Fabian said, he's welcoming questions at the moment. So if anyone else has questions, please feel free to put them in the chat or just go ahead and unmute yourself and ask.
To state the obvious, reach out if you have questions. We'd love to help. And this is not just for NCI data. This tooling can be used for your own data and comparing it against NCI data sets if you like, or other tooling. So it is broadly available.
Hopefully I didn't overwhelm everyone.